So I'll start this talk, as I start every talk I give, actually, with some personal background and a photo from my wedding. This is me getting married in 2009. That's Eric Minical, my husband. It was a gorgeous day in August, and that's a photo of my mom helping me get ready that day. So she was 51 years old, and she was healthy, and she was sort of the life force behind the party. She organized everything. She was beaming from ear to ear. But this is the last family event for which my mom was actually healthy and happy and with us. At the beginning of 2010, she started to experience what seemed at the time like some minor symptoms. Vision was blurry, some unexplained weight loss, and this very, very quickly snowballed into what I can only call a catastrophic neurological decline. In about 4 or 6 weeks, she had descended into a dementia where she could not communicate with us, recognize us, live independently. And about a month after that, she was on life support. We raced around the country. We were looking for a diagnosis, for any information on what was happening, but we actually never in her lifetime found one. She passed away at the end of 2010. And it was only from her autopsy that we learned that she had died of genetic prion disease. And embedded in that piece of information was this word genetic, and the idea that out of the clear blue sky, I was at 50-50 risk of having inherited the variant that had done this to my mom. So that was a tough time for us newlyweds. We talked to a lot of different people. We talked to friends, we talked to family, we talked to physicians, genetic counselors. And I would say, overall, if I had to sum up the advice we were given, we were basically told, take this opportunity to walk away. There's nothing you can do to treat or prevent this disease. An unlucky result is something that you can never unknow once you know it, and there's just nothing you can do with it. Nevertheless, I think we weren't really wired to walk away. I knew about myself that I couldn't go back to a time before I knew I was at risk, and so I would wake up in the morning flipping that coin in the head for the rest of my life, either way. So might as well know what I was up against, give us that 50-50 shot at knowing that I was safe, and if not, it couldn't be worse than it already was. So we got the test, and it was here locally at the Brigham, end of 2011, that we learned that I did inherit my mom's variant. So long story short of what this means for us, for me, this mutation causes a fatal, untreatable neurodegenerative disease. My variant, and I'll show you sort of a deeper dive on this in a moment, is highly penetrant, so near certainty of developing disease at some point in my life. An interesting thing about prion disease is that we can't say when it will happen, so it could strike at any point in adulthood. But once it does, after a variable number of years or decades of being perfectly healthy, you go into, as we saw with my mom, a really, really steep symptomatic decline. And usually from first symptoms to death is under a year. So that was quite heavy. And to give you some context for how we were hearing all of this, I had recently graduated from law school. Eric was trained as a transportation engineer. We did not have biology backgrounds. So we were sort of hearing these words, taking them at face value, and then going home and Googling what we could, and trying to build a little bit of context for this enormous change in our lives. So didn't have biology backgrounds, but did have access to the internet. And that proved to be a pretty good launching pad, actually. And what we learned as we were Googling around was that we had basically a bottomless amount of curiosity, bottomless motivation to learn more about this rare disease. And it ended up, this news ended up catalyzing a change in our lives that I think neither of us could have possibly in a million years predicted. And we owe this in part to the fact that we already lived in this amazing town. But after getting that genetic test report, we both signed up for night classes locally. I started walking into classes at MIT. I think our initial motivation was we just have to learn enough biology that we can be good advocates. We can follow the research in this field and see if something comes along. But it took on a lot of momentum, and soon that was what we wanted to be thinking about all day. We ended up leaving our jobs, and I took a job as a tech in a genetics lab at Mass General. This was just about 3 months after getting that test report. And I was very much it was sort of a leap into thin air, transitioning to the absolute bottom rung in an absolute brand new field. But it felt like the only sort of productive frame we could put on things, like a positive way to engage with this new information. And I had a lot of energy for it. So Eric followed not long after to actually a bioinformatics role at MGH. We were there for 2 years, and then we both started our PhDs at Harvard Medical School in 2014. And through the search for a home lab, we found Broad. And I would have to say, being based here has really been sort of a watershed for us, because Broad has invested in our mission and really empowered us to spend these years, these so-called trainee years, pursuing our mission and doing exactly what we need to do to try to move on this disease. So it's now, I guess, 7 years on, and we just defended our PhDs. We're now launching our own independent lab here, and very much full steam ahead. I think we're far from the end of this quest. We're really far from the end, but I think we're also pretty far from the beginning. So that's gratifying. And after 7 years, I think what we can show for ourselves is that we have a therapeutic strategy that we really believe in. We have a drug development program with a major pharma partner that we're working to move forward every day. We have a regulatory strategy that I'll tell you more about, that's not business as usual. And so we're working closely with FDA to try to make sure we're paving the right path. And we have a motivated cohort of people with test reports like mine, or people at risk who lost a parent the way I did. And very much trying to build a team there, because we definitely can't do it alone. So I'll tell you a little bit about our work, and the frame I want to put on it is we're like card-carrying scientists now or whatever, but I think very much our work every day is informed by the fact that we're patient scientists, not just scientists. And I think that informs how we do the work, but also like what work we think it's important to do. And the deeper we get into it, the more I'm reminded that that like just orients us differently from a lot of people we know every single day. So I'll talk a little bit about quantifying risk. Prevention is a huge thing for us. Practicality, trying to identify a solution that's not just elegant, doesn't just sound good, but could actually work in our lifetimes. Getting the information out as we generate it, and trying to rally the troops. So quantifying risk, you can imagine when we were first handed this whole idea, we were obsessed with the idea of what kind of risk was I at, what numbers could we put on it. Of course, before I was tested, it was 50-50 for an autosomal dominant disease. But then once we got the report back, we found that we weren't totally satisfied that we had the answer. Was the answer now, yes, 100% of the time, because you have this variant, you will develop this disease? And this turned out to be like a question that we could really sink our teeth into once we transitioned over into research jobs. Because as I'm sure you guys have been talking a lot about in the past couple days, we can't necessarily take the literature at face value. There are lots of reportedly pathogenic variants in the gene that causes my disease, PRNP. And the evidence supporting those variants varies a lot. And in the meantime, this is not unique to our disease gene, right? There's a lot out there in the literature. There's a lot of pollution. There's no self-correcting mechanism. A lot of it just never goes away. So one of the very first questions that we embarked on under the mentorship of Daniel MacArthur here at Broad was the question of how certain are we about these variants? What can we say about the variants in this gene that have been reported to cause disease, including mine? So what we had access to at the time were population controls in the form of the EXACT database. And it now looks like a quaint number of population exomes of 60,000. So we weren't yet at the scale of hundreds of thousands of population genomes. But this was by far the largest data set of its kind. And we also had a collaboration with 23andMe, where we were able to look at SNP data from people who had opted into research. So we could sort of look in an unbiased way at population controls. And we also set about gathering a lot of case series from different prion disease surveillance centers around the world in the hope of sort of comparing those two data sets. And the sort of overall, principle, I guess, informing how we were looking at these numbers was this idea that if these variants are completely penetrant, then when we look in the population, they need to be as rare as the disease. If they're not, then that tells us that something is fishy. And it turns out that they weren't as rare as they should have been. So when we looked at EXAC, and similarly when we looked in the 23andMe data, we saw many more, like in EXAC it was 30 times more PRNP, so-called pathogenic variants, than we should have based on how often we see prion disease. So basically, what we were able to do with these population controls on the one hand, case reports on the other hand, was to ask how often do we see these variants in these two groups? And what fell out of this was some variants, and it's the ones with really good pre-existing evidence for pathogenicity, like old-fashioned family trees where 50% of people develop disease, or someone's modeled that variant in a mouse, and the mouse reliably develops disease. So these variants we saw in cases, and we did not see in controls. It was a pretty clear story. Conversely, there were variants that sort of popped up in the literature here and there, but there had never been a closely reported case series from a family. There had never been a mouse. And a lot of these variants we saw were rare in cases, common in controls, and could very well be benign. And then there were a few in the middle. And the story here wasn't consistent with either extreme. Probably don't cause disease 100% of the time, but probably not completely benign. So these might be risk factors. And what emerged from this was the realization that not all variants in this disease gene are created equal, and there's actually a spectrum of risk that they convey, as kind of shown here. So what came out of this was encouraging news for some people. And actually, one of the direct results of this study is that people who we know of, who carry some of these variants, who had been walking around thinking that they were at the same risk that I am, were called up and recounseled and told, actually, that variant is probably low risk or benign. You are probably not going to die of prion disease, which I think is amazing. That was not the case for all the variants. It was not the case for my variant. And we hadn't really gone in hoping that this was going to make all our problems go away, because there was very good historical evidence that my variant probably was highly penetrant, and that turns out to be true. So I think this mostly just reinforced the mission that we'd sort of already embraced, which is we have to develop a drug. We have to develop a treatment. And in particular, segueing into our second mandate, we have to develop a preventive treatment. So I already talked to you about the timeline of my mom's illness. On paper, it was 10 months long. But if you drill down on what this year looked like in my family's life, those first symptoms in February were very minor. No one was saying, oh my gosh, this is the beginning of a fatal neurodegenerative disease. And by the time we get into March, April, already her ability to conduct her activities of daily living independently was gone. Her quality of life was gone. You can see she was on a feeding tube and ventilator by June. So I think saying it was 10 months long really overstates the case. A lot of this time was on life support, and the window in which meaningful quality of life could have been preserved was really weeks, if existent at all. So this is a really, really rapid disease. And I think that has implications for how we need to treat it. So this is the genetic blueprint of prion disease. We have a single gene. We're so lucky to be up against a single gene problem. The prion protein gene gives rise to one RNA, which gives rise to one protein. And we actually all have the normal prion protein. It's not normally a problem, but it's capable of misfolding into this pathogenic form that can go around grabbing other molecules of prion protein and conformationally converting them so that you get this exponential cascade of prions through the brain that kills neurons. That's the disease in a nutshell. And what was interesting to us looking around the field, prion disease has been around for a long time. People have been studying it since the 80s. But drug development efforts before we came along had really been focused on either misfolded prions themselves, because that's kind of the bad guy, right? Or cell death. This idea of, can we surround the neurons with helper molecules and protect them? Okay. There's a lot of interesting science in this part of the disease process. There's a lot of unknowns. But these elements of the disease are only present when you're already on that downhill, right? This is the symptomatic phase of disease right here. And as we thought about what is our mission, what do we want a drug to do in my body, we don't want to wait, sit on our advance notice, thanks to my test report, and just wait and wait and wait, and then try to do something at this stage. What we need to do is get in ahead of time. Our mandate is to prevent disease. And I think that means going after molecules that are present before that's happening. It's like a really clear shift in how we have to think about the molecular job of our drug. It turns out that we have really good pre-existing evidence from transgenic animals that the more prion protein an animal has, the faster it develops disease after you infect it with prions. And the less prion protein it has, the longer it takes to develop disease. And an animal that has no prion protein at all never develops prion disease. And in the meantime, there's actually a wide variety of different knockout models, both engineered and naturally occurring, all of which are healthy. And one additional finding that fell out of that penetrance study that I described to you, in the exact database, we were able to identify for the first time people who are walking around with one loss of function allele for the prion protein gene. So they're not total knockouts, but they probably have 50% reduced gene and protein dosage, and these people are healthy. So I think all of this gives us some information that reducing the amount of normal prion protein should be an effective strategy against this disease, and it actually should be safe. And one additional lens on whether it could be, whether it's safe to reduce the amount of this protein. So we can sort of look genome-wide at how tolerant different genes are to loss of function mutations arising. On the far end of the scale, topoisomerase 1, for example, if we look now in the Nomad database across hundreds of thousands of population genomes, we don't see any loss of function variants in that gene. So it is intolerant to reduced gene dosage, potentially. There's natural selection against that. There's a whole spectrum in between, and it turns out that the prion protein gene basically anchors the other end of the spectrum, where when we look in Nomad today, we see just as many loss of function variants as we would expect to based on chance, based on the size of the gene and the basal DNA mutation rates that we expect. So I think that gives us sort of an additional, maybe little vote of confidence that this could be a drug target to pursue. So basically, our mission turns into this. We need to target either the prion protein or one of its precursors and reduce its levels. So how can we do this super practically? How can we do this in a way that's going to make a difference for us and for other people living with these mutations today? We've been super lucky to have amazing mentors along the way. And one of them is this guy. And I think he understands our urgency better than anyone. This is Jeff Carroll. He's a Huntington's disease researcher who carries the Huntington's disease mutation. And I don't think it's a coincidence that he was the first person to say to us, have you thought about antisense oligonucleotides? So some of you may be familiar with ASOs, They've been around a long time, so these have been explored as a drug modality since the 1990s. They're not sexy, they're not new. Basically, they're small modified pieces of DNA, chemically modified for stability, and you can program in a sequence of nucleotides to target a specific RNA. And then that causes the ASO to bind that RNA, and out of that binding event, you can get various mechanisms of action, but one of them is that you can trigger that RNA to be degraded. So could this be a really specific way to reduce just this one RNA that we don't like that encodes the prion protein? Things that were exciting to us about ASOs, and this isn't necessarily the stuff that makes a really cool, sexy paper, right? But the fact that ASOs had been around for so long meant that a lot of information has been accumulated about how they behave in the central nervous system. So we actually inherited a lot of information about dosing and distribution and washout and tolerability. And the fact of these drugs not being novel is actually exactly what made us say, this is something we can probably advance on the timeline that matters to us. This is something we can get to patients. And the other thing that gave us confidence, and part of where all that great data comes from, is that ASOs are in humans for a bunch of brain diseases. So you may be familiar with the story of nusinersen, which was the first ASO approved for a neurological indication. Spinal muscular atrophy, it's like just a horrifying genetic disease that in its most severe form kills kids before the age of 2. And at this point, if they get this drug, it is capable of stabilizing those kids, or if they get it before onset, so far, from what we see, capable of preventing the symptoms from happening. So I think this has been an extraordinary success story. Huntington's disease is just a few steps behind. There's a phase 3 that has just recently launched for an ASO to reduce the amount of Huntington protein in the brain. And that is like a really close kind of parallel for what we need to do and a program that we're trying to piggyback off of. So to sum up, like 4 years of work in one slide, we've been collaborating with Ionis Pharmaceuticals on ASOs to reduce prion protein in the brain. And there are so many survival curves at this point, but I'll just show you one because I think it kind of sums up the story. These are animals that got 1 dose of ASO before prion infection, 1 redose 90 days later. And you can see that the treated animals in blue and green outlived their untreated counterparts by 60 to 80%. So this was with 2 doses, and we know that the drug washes out after 90 days. So I actually think this is a lower bound on efficacy, and a super exciting result for us. And not captured in the survival curve is the fact that by monitoring symptoms and nest building and weight loss and all sorts of other things, what we saw is this isn't just an extension of survival per se, it's an extension of healthy life. So I think this is what we're in it for. Lots of work still to do, but we felt comfortable taking this project public last year, getting up in front of a room, pictured there at the bottom, of patients and families and telling them that this project is in development. And the reception has been super warm, and it's nerve-wracking. And I think we set a really high bar for ourselves in terms of what we're willing to speak to the patient community about. I think we're so conscious of not wanting to spread false hope and of all the uncertainties and all of the work that's still ahead for this program. But we felt like we were at a point where the responsible thing was to share it because we need people to be thinking about whether they want to be involved. And people need time to mull that over. So we did announce the program, and I'm so glad it got the reception that it did, both from the patient community and sort of the general public. I'll just highlight briefly that that's in dire contrast to how this work is received sort of in the system. We're getting manuscript and grant reviews all the time that remind us, as if we didn't know, that ASOs aren't novel, and like to sort of poke at the fact that prevention, well, okay, but what's more impressive is if you throw yourself into the car wreck as it's happening and show that you can make a difference then. So I knew when we got into this that we were going to be fighting this disease for the rest of our lives. I don't think I realized just the extent to which we were signing up to fight the system. But I guess life is surprising, so that turns out to be part of our mission too, I guess. But I feel really strongly that this is our mandate, so we'll keep doing what we can. Getting the information out. So I'll pivot to a different arm of our work, which is natural history, looking at genetic prion disease mutation carriers. And as we're sort of building this community, one larger backdrop to this work is that in other neurodegenerative diseases over the past 10, 15 years, this narrative has emerged that the disease doesn't actually start when you thought it did. It doesn't start the first day you forget your keys. Actually, for 5, 10, 15, 20, 25 years before that, potentially in the case of, say, inherited Alzheimer's disease, there was all this underground stuff going on. There were all of these detectable changes, be they molecular, in your blood, in your CSF, or sort of structural in your brain that you could see with a PET scan or MRI scan. This has received a humongous amount of press. I remember reading about it in Time magazine a long time before we ever became scientists. So I think that has sort of set a whole background of expectations for what neurodegeneration means. With that in mind, we launched into this MGH clinical study that we set up at Mass General with Steve Arnold, our neurologist collaborator who's been amazing, with the idea of bringing in people with genetic prion disease mutations or people at risk for them and controls, and having them donate biofluids so that we could start doing some characterization on our potential trial population of interest. One thing we've learned along the way is that this population is really motivated, which was really cool to learn, and I hope that bodes well. So we've had really good sort of participation. People come in, they do a battery of different tests. The big thing for us is the lumbar puncture. And CSF samples come to our prion lab here at Broad. And I'll show you a little bit of the data that we've generated so far. So two big things that we've been looking at, and anyone who thinks about brain disease is super familiar with these markers. Total tau and neurofilament light chain have emerged as these like really early sort of canaries in the coal mine, proteins that with levels that rise in the course of neurodegenerative disease as neurons die. You can see our positive controls, these are people with symptomatic prion disease, have dramatically elevated levels of both of these proteins. But when we look at our non-carrier controls, and when we look at our carriers of various different prion disease-causing mutations, everybody clusters together. Everybody looks about the same, and everybody falls in the normal range. And as we're following people over time on the bottom there, this is preliminary. We're just starting to bring people back for annual follow-ups now. But so far, we don't see any sort of upward trend that looks fishy either. It actually just looks like carriers appear healthy. So this is a negative result. And people are, typically not super excited about negative results. But I want to argue to you that negative results can be a really big deal. I think it's actually really meaningful to the carriers who we're asking for their time, we're asking for their trust, we're asking for their biofluids. I think it's really meaningful to be sharing back the information that what we see is that they appear healthy. And I think this actually has implications for how they see themselves, how they think about their own bodies, how they interact with their doctors, their medical care. And sadly, in the case of genetic prion disease, this is actually a community that has faced and still faces a lot of discrimination in the medical establishment. And we hear routinely about people being denied routine medical care, routine procedures, because of the fear that these carriers are prion contaminated in some way. So I think starting to amass evidence that these folks are healthy is a huge deal and can potentially push back on some of this. So I'm very much going to be pushing for this negative result to get out there, to get into the world. I think there's a second way in which it's important, which is in diseases like Alzheimer's, people are thinking about something called secondary prevention. This idea that if we have all these changes that are occurring under the surface for years, why don't we track those? Why don't we use those in trials? We can treat towards those changes. We can use them as endpoints. So we can use that prodromal phase to our advantage. If we don't have a prodromal phase that we can point to, I think it's time for us to stop assuming that it's there. I think this is an innocent until proven guilty type situation. We have to be preparing to move forward without it. And for us, that means setting up infrastructure that can support primary prevention. So based on nothing but genetic diagnosis, getting a treatment to people to reduce their risk and keeping healthy people healthy. So one very great challenge in that domain is building our team. We have 60 plus people who have either participated in the Mass General study or are signed up to participate. I think that's really great. In some other disease, that might not sound like a lot, but we've been really heartened by that. But that's the tip of the iceberg. We think if we were counting everyone with a mutation like mine, whether they know about it or not, there are probably low thousands in the United States. And we need to be finding these folks. They have an opportunity right now to participate in research if they want to that actually tangibly moves our drug development program forward. They may have an opportunity in a couple years to be part of a trial. They may have an opportunity in a few years to take a drug that reduces their risk. This is all quite actionable, right? This is actionable in the short term. And yet, I know a lot of these people are getting counseled the way I did, to walk away from their risk, walk away from the chance to know. Don't get tested, there's nothing you can do. What can we do to change that conversation? I'm not sure. We're doing everything we can. We are reaching out through the community. We're taking every opportunity to give talks to people who interface medically with this community. We have launched an online registry. I think at this point we have 130 people who identify as healthy but at risk. So slowly we're pushing these numbers. But we're soon going to get to the point where we actually are in a position to try to start helping people, and we can only help the people we can reach. So I think to the extent that we can reorient this conversation and kind of really, really push on this idea that there's nothing you can do, and get people to see this a little differently, it'll make a huge difference. So where do we go from here? In the next few years, I hope that a PRP-lowering ASO will reach a trial in pre-symptomatic genetic prion disease mutation carriers. There's a lot of unknowns still, but we're pushing every day to try to get there. Within the next few years, I would like for us to have some idea of what the second PRP-lowering drug could be. Maybe one that doesn't need to be dosed as often, or that does a better job of reaching all brain structures equally. I think there's a lot of room for improvement. An ASO won't be a cure. And now we've built infrastructure that can support that second drug to, I hope, advance more swiftly. The big picture beyond my one rare disease, I think we are super passionate about applying what we've learned to move forward a broader conversation about prevention. Prevention of brain disease, prevention of genetic disease. These test reports, my test report, other test reports like it, are giving us advance notice that we can't afford to squander. And I think we are working hard to figure out how do we do rigorous trials in people who aren't sick yet. And I think in diseases where we have a fair amount of genetic clarity, where we have good molecular biomarkers, right, where we have the ability to define a patient population, we have a strong therapeutic hypothesis, and we can read out the results with a biomarker we trust, I think this can be done. And I hope that we're going to be a little bit of a pilot case in my one funny rare disease where we don't have a lot of other good options. I hope we can try to lead the way. And slowly, just by being part of both worlds, I hope we can try to bring these two worlds a little closer together in terms of the things that are important to patients, like getting there fast, and the things that biomedical research is really set up to reward. A question that we are sort of increasingly grappling with as we spend more time in this world and connect with more people and start getting more and more phone calls and emails from other people who've gotten a rare disease diagnosis for themselves, for their child, is what can we do to help support fellow travelers? Besides, I hope, be an example of people who have helped get a drug program off the ground. I think there's a lot that we share with these folks. And if people are reaching out to us, it's already kind of a given that they were information seekers just like we were. They felt really passionately that they wanted to know what was up with their body or their child's body, and they wanted to pursue that answer as far as it could go, and then maybe they want to push it further than it can yet go. And I identify with that so much, and with the idea that actionability isn't a static thing, it isn't a binary thing, and it isn't really a thing that someone can tell you. It's a story that you're part of. But in the weeds, it's really complicated. And there isn't sort of like one answer that we can give people. And I think this is something we're still grappling with every day because the details matter so much. And it's partly, what are the genetics of your disease and how much do we know? And how does it present and when? And where do you have to get a drug? And what does that drug have to do? And are there existing platform technologies that could address this problem today, but for the resources and the connections? Or is this a problem that actually has to be solved from scratch? And what resources exist, right? Have other people started building some of this infrastructure? We already had a great biobank in prion disease. There were already 2 patient organizations. A lot of diseases aren't so lucky. So I think maybe a theme that'll be familiar to you folks too, and certainly something we think about all the time, is how to convey the right amount of hope in these conversations. Not too much, not so much that it's irresponsible, but I also think we have to be worried on the other side, that it can be irresponsible to convey too little hope. So hard balance to strike. Final thought about prevention. This is my daughter, Daruka. So she was born in July 2017, and we had her through IVF with preimplantation genetic diagnosis. So I used to end talks with a photo of Daruka, and I used to treat it as just a real high note and be like, we did IVF PGD, and it worked, and it was great. So yes, there's a lot you can do with your genetic information, even today, even without a drug on the shelf. And I believe all of that. It is great. It did work. It was also so hard. And I wonder sometimes if IVF PGD is the hardest thing I've ever done. I think for every success we see out of the fertility medicine industry, there is just such an untold amount of failure. And for us, the failures I can actually quantify for you. Failures to successes to date, 10 to 1. But we got the 1. Right? So I think to the extent that your life and work touches on these themes, I think IVF PGD is amazing. I absolutely talk it up to everyone in the genetic prion disease community as an amazing option that's out there, and it's a miracle that it ever does work. But it's for sure a hard-won miracle. So. With that, thanks, and I'm happy to take some questions if we have time.
I just wanted to get your thoughts. So I am very preventive genomics, but I was just at a Harvard course, ethics course actually last week, and they were saying how they think preventive testing is bad, not just for genetics, but many things. And so I just wanted to get your thoughts. Their main point is, you're stressing out patients, there's not a lot they can do, but I think your story just proves that wrong. But what do you say to those people in those situations to help them see the power of information?
Yeah, it's so hard, and I think I always lead with everyone's different, and with, I think, something as sensitive and as personal as predictive genetic testing, as much as people choosing to get tested is an essential element of, what we can move forward on behalf of the whole community, I try to always make sure to tell, the community when I speak to them, it's up to you. And it's a huge part of it is knowing yourself. We knew instantly that we were need-to-know kind of people. I think a lot of people know instantly that they actually do have that superpower where they can put the fact that they're at risk away in an attic of their mind and not visit it and live a normal life, without it. And good for them. What I worry about, though, is that there's a lot of people caught in a really terrible limbo where they know they're at risk and they're thinking about it a lot, but they've received advice that they shouldn't pursue it, and so they're constantly fighting this tug of war in their mind, and they can't put it down. And I can tell you that was me in the absolutely torturous 8 weeks between learning I was at risk and getting my predictive test report. I woke up every single day thinking about, is it yes, is it no, is it yes, is it no, is it yes, is it no? And I think there's actually a whole lot of research on this in psychology, like adaptation and the idea that once you have the information, even if it's not what you hoped for, you begin to adapt to it. It just becomes the furniture in your mind, right? But if you don't know, you can never have that. You can never begin that process. And I worry we're doing a disservice to all those people in the middle by counseling so conservatively. I also think that we're not the only rare disease where a few or one outsized sort of motivated patient has moved the ball in whatever form. And maybe it's not that you go develop a drug, but maybe it's that you become an advocate or a funder or the person who organizes the patient community or the person who launches a biobank. And I think when we look around us at the world, which rare diseases now have prospects for treatment? It's the ones where those people had the information, right, and had the motivation to step forward. So I think to issue a blanket statement that everyone should get tested is irresponsible. To issue a blanket statement that no one should get tested, I don't see how that's any less irresponsible. We have to give those people a chance to do what it is that's actually going to bring them peace. And maybe it's not hiding, right?
Thanks. So I loved hearing your patient scientist story, and I think we're all patients in some level, although certainly some people have a stronger connection to a particular gene or disease. I'm a genetic counselor by training, and I think as clinicians, we're kind of trained to keep our personal outside of our patient interactions or things like that. So I'm kind of curious to hear your thoughts on clinicians and scientists being more public with their personal connection to disease, either with specific patients or just in the general conversation surrounding disease.
Yeah, great question and great point, for sure. Everybody has, the perspective of being a patient, and it's just a question of how directly that interfaces with your work. It's interesting. It's a thing that we got a lot of pushback on in the beginning. And one particular form that pushback took that I still really object to, I did at the time and I still do, is the idea that because we're personally affected, we couldn't be objective. And that somehow we were going to be compromised. We were going to be too hopeful or something and chasing down the wrong stuff and unable to handle negative data and unable to switch course. And when I think about the past 7 years, I think it has so profoundly been the opposite. In fact, I think no one is more motivated to discard anything that's not working and pivot to a thing that is working than we are. And that it actually is another way in which we're in constant tension with the biomedical establishment, is that even pursuing something to the point of nominal completion, to the point where you're writing up the paper or you're helping your trainee sort of wrap up that project and advance to the next stage, all of these things are in tension for us with dropping a thing the moment it is not going to be a drug in our lifetime, which we feel is our mandate to honor the patients who are looking to us. So I think, if anything, I think that we have all our incentives aligned to be objective. And I don't know. I think our science reflects that. You guys tell me. Yeah. What else about being a patient? I think over time we found that a lot of people are responsive to the idea that you can be both. And a lot of people have actually shared with us, like, I think, who don't necessarily share it publicly, the personal thing that got them into biomedicine in the first place. And yeah, I hope that like some of the stigma around that will go down over time, because I actually think that like being in touch with your inner patient is like a really important sort of North Star, like a way to stay oriented in a profession that, like every profession, has a really, really complicated web of incentives that sort of govern the directions people go in and the things they value and the things they do with their time.
That was fantastic. Thank you for sharing your story. I have a question about the trial that you conducted at Mass General in terms of whether or not, and there's an underlying reason I'm asking this, whether or not people underwent genetic testing in order to participate in the trial. Because it's my observation over time that the minute there's even something you can do, including participating in a non-therapeutic trial, that pushes some people to go ahead and get information.
Yeah, great question. So I'm always super careful to call it a clinical research study. They're not letting us use the word trial, I think appropriately, because we're not giving anybody a drug. But the way we set up that study, and we thought about this, and our decision was, let's take everybody at risk, and we like set out some criteria for what that would mean. But basically, first-degree relative who died of confirmed genetic prion disease, or two relatives within a certain distance who died of prion disease, not confirmed genetic, but quite likely genetic. Or if you've been tested yourself, people are certainly eligible to enroll. And one of the functional consequences of setting it up that way is that many people who enroll are at 50-50 risk. And the ones who test negative end up being our sort of basically perfectly matched negative controls. And so in that way, we're sort of building out both populations at the same time, which I think is going to be important, in the details of being able to compare these samples. But I also, we wanted to be inclusive in the sense that we're building relationships. Some of these people are untested. Some of them ultimately will test negative. But if they're motivated to participate, if they're motivated to be involved in the community, potentially they are like a node, right? They're connected to other people. They're connected to their relatives. They're connected to other people who might someday want to receive the drug, and to whom it might be meaningful that there's this study. So I think we wanted to cast a broad net. When we think about a trial, I do think people are going to have to be tested, because probably an ASO will be intrathecally delivered, and even beyond that, I think the bar for giving an experimental drug to people is just going to risk-benefit is going to demand it. So I think we also the idea of staging it in that way, sort of giving people a broader opportunity to participate and this time to think it over, and then coming back to them in a few years and saying, now we're actually ready for the next step, and we're ready for the experimental drug to hit the clinic. But you've had this time to think it over. Now we do need to ask that you get tested if you want to participate. I hope that staging will appeal to some people.
So I'm not sure I'm understanding then. People who participated in the clinical study could have been at risk but not have risk. Were at risk because of an unknown genetic but family history, correct?
Right.
So, and then you knew their genetics when you analyzed the data? Is that correct?
We, as the study team here at Broad, see only de-identified samples. They're all coded, and then we see the data results. The study team sees only the people.
And they're not connected.
And they're not connected.
Got it.
That's
Got
right.
it. And then a follow-up question about the potential clinical trial is, and not to be a naysayer, but what's the endpoint, the efficacy endpoint?
Yeah. So I didn't go into this in detail, but basically what we're proposing is that prion protein is the only essential protein for this disease. So it's directly on pathway. And there's, as I showed you guys briefly, outstanding evidence from genetic models that if we lower the amount, that should be protective. So we've been working on characterizing prion protein in cerebrospinal fluid, just the normal protein. And we know that it's abundant, it's detectable. If we do everything by protocol, if we're careful, we can measure it reliably. It's coming from the brain. And from our data so far, I think there's every reason to believe that if we modulate the amount of PRP in the brain with an ASO, we can read that out in a reduction in prion protein in CSF. So we're launching now rat and monkey studies to confirm that in a preclinical setting. But so far, our conversations with FDA have been about the idea of using PRP and CSF as a so-called surrogate endpoint, with the idea that we could get potentially provisional approval just based on moving that biomarker that is reasonably likely to predict clinical benefit, and then follow people long-term to confirm clinical benefit once the drug hits the market and is available to everybody. So that's to me, the dream scenario where we don't randomize anyone to placebo and then watch them die. We'll see.
And then, so not brain biopsy or something more invasive?
Nope. Because I actually think CSF is a better readout than a brain biopsy because it's a whole brain disease. So we need to reach as much of the tissue as we can. And a biopsy, like, you'll get one region, and potentially you'll get a cortical region, you'll get something that's easy to access. And we know that the distribution of ASO uptake is different in cortex versus other brain substructures. So I think a bulk readout, I think CSF is actually a good sampling compartment in that sense. Yeah.
Interesting. Great.